non-carbohydrate sources. Now, when I say non-carbohydrate sources, it means proteins or lipids, but lipids cannot be utilized in our system for synthesis of glucose. So, it is a protein or amino acids, or more precisely the glucogenic amino acids, which leads to formation of glucose. So, it is the glucogenic amino acids which leads to the formation of glucose. Amino acids are produced by the protein catabolism. Muscle protein breakdown when there is a scarcity of glucose in the blood, when mostly it is during the night time when we are asleep. When we are sleeping, we are not eating anything. So, there is no continuous supply of glucose to replenish the glycogen store in our liver. Our liver stores glycogen for supply of glucose for the future use. But when we are sleeping for more than 4 to 6 hours, that liver glycogen gets depleted, it gets over, finished. But after that, the glucose is supplied by the breakdown of mostly muscle protein. If we do not eat further, then small amount of hydrolysis of fat or triglyceride will take place, from where a part of it, glycerol. Glycerol is nothing but a derived lipid. So, glycerol, though being part of lipid, may participate in synthesis of glucose. Now, the synthesis of glucose mainly based on this pathway, gluconeogenesis, that is almost reversal of glycolytic pathway. Now, if you can recall the glycolytic pathway, there are 3 irreversible steps. So, theoretically, if we can reverse those 3 irreversible steps, then we can synthesize glucose from pyruvate. But problem arises that for synthesis of pyruvate, glucose and pyruvate both are available in cytosol. But for synthesis of glucose, the other way around, pyruvate is available inside mitochondria, not in cytosol. So, this is the first hurdle. What is the first hurdle? To bring pyruvate from mitochondria to cytosol. To do so and to bypass the third irreversible step of glycolysis, that is catalyzed by pyruvate kinase, it takes 2 enzymes, Pyruvate Carboxylase and PEPCK, PEPCK, phosphoenol pyruvate carboxykinase. This one, pyruvate to oxaloacetate by pyruvate carboxylase, and then phosphoenol pyruvate carboxykinase. This enzyme requires GTP, right? So, there is no question of having scarcity of ATP for ATP production, right? Now, these 2 reactions, though looks like seemingly very timid reactions, but they do not take place like that. Why? Because this oxaloacetate is produced inside mitochondria. Then it is converted to malate via malate dehydrogenase. Because oxaloacetate has no transporter for transport across the mitochondrial membrane. So, it has to be converted to malate. The moment malate is formed, now malate has specific transporter for transport across the membrane. How? Malate aspartate shuttle. So, it comes out outside. Outside it forms again aspartate, aspartate to malate, malate to oxaloacetate. Now, all these reactions are complementary. So, those reactions we do not consider here, just writing these 2 reactions. You see this one here, we will come to that, the reactions are there. Okay, for the timing, now see what are the organs involved for gluconeogenesis, liver and kidney, and very little in brain. Now the question is, who needs this glucose? Who needs this glucose? Which tissue? Which tissue cannot survive other than glucose? Cannot survive. Without glucose, it cannot survive. RBC. Mature RBC cannot survive except or without glucose. Pyruvate carboxylase forms oxaloacetate. This reaction requires 1 molecule of ATP. Correct? Second reaction requires 1 GTP. 1 GTP is equivalent to 1 ATP. So, the pyruvate kinase step produces 2 ATP, but conversion to phosphoryl pyruvate requires 4 ATP. So, this is seemingly energy wasting pathway, but still it operates. Why? Because, last question, there are few tissue which cannot survive without glucose. Even if you give them lot of glucose, RBC will perform anaerobic glycolysis, but it requires only glucose to survive. Now, these two reactions will produce pyruvate to phosphoryl pyruvate, oxaloacetate convert to malate, and malate comes out, malate to again oxaloacetate. Now, this is cytosolic MDH, this is mitosolic MDH, mitosolic and cytosolic. These two are opposite. The process by which malate comes out is against one aspartate via malate aspartate shuttle. Correct? Phosphoryl pyruvate. Now, from here we can go up to fructose 1,6 bisphosphate because all the reactions are reversible. From fructose 1,6 bisphosphate we require 1 bisphosphatase enzyme to form fructose 6-phosphate, then phosphorus isomerase will convert it to glucose 6-phosphate and then glucose 6-phosphatase to form ultimately glucose. Right? Is this the only way? Suppose there is formation of lactate, correct? The lactate, see, when you synthesize lactate is synthesized outside mitochondria. So, it is outside mitochondria, or you can say where there is no mitochondria there is lactate, like in mature RBC. Again, where mitochondria is there but still lactate is produced, where? Skeletal muscle. So, whatever may be the way, lactate will be produced outside mitochondria. Lactate is converted to pyruvate which enters mitochondria for further reaction. Now, inside this will convert to oxaloacetate, phosphoryl pyruvate and follow the same route. So, two sources, one is lactate, another is pyruvate. How they are going to the phosphoryl pyruvate is shown here, right? While if it comes separately, you have to write this pathway in detail. If it comes like a big question, then you might omit these two steps. Okay, just mention 2 pyruvate carboxylase and phosphoryl pyruvate carboxykinase. These 2 steps only sufficient. Third source, glycerol. There are many other books which have given it little differently. You can choose any one, doesn't matter. Glycerol is first converted to glycerol 3-phosphate by glycerokinase, then glycerol 3-phosphate is converted to dihydroxyacetone phosphate. Dihydroxyacetone phosphate is by dehydrogenase enzyme. Alcohol to ketone. DHAP or dihydroxyacetone phosphate is one of the intermediate of glycolysis, right? So from there it can follow up to the glucose, but it has to cross only two more irreversible steps: fructose 1,6-bisphosphatase and glucose 6-phosphatase. So always remember these four enzymes, you cannot forget. The question is very easy from here. Name the key enzymes of gluconeogenesis. Four enzymes you remember: pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase and glucose 6-phosphatase. কি বললাম বলো? Pyruvate carboxylase, PC, PEPCK, fructose 1,6-bisphosphatase and glucose 6-phosphatase. Glucose 6-phosphatase. Right. So glycerol can give rise to glucose through this. There are other substances, like I mentioned, glucogenic amino acid. Can you enumerate glucogenic amino acid? What are glucogenic amino acids? বলো, name glucogenic amino acid. Again বলো. Anyone? Correct? Hearing? All these give rise to, দেখো লাস্ট ক্লাসে দেখো একটা লেখা আছে না বড় চক্র করে, ওখানে যাও দেখো, গ্লাইসিন, অ্যালানিন, সেরিন, থ্রিওনিন, সিস্টিন অ্যান্ড ট্রিপ্টোফ্যান। ট্রিপ্টোফ্যানের দুটো পার্ট থাকে, ট্রিপ্টোফ্যানের একটা পার্ট থেকে এখানে আসে। ট্রিপ্টোফ্যান থেকে প্রথমে অ্যালানিন তৈরি হয়, সে অ্যালানিন থেকে পাইরুভেট তৈরি হয়। Once they get into pyruvate, then they can follow that previous route. Clear? If they are from other substances, like say for example, odd chain fatty acid. When odd chain fatty acid is oxidized, it produces alright, it produces number of acetyl coenzyme and one propionyl CoA. Now, what is the source of propionyl? What is the route of entry of propionyl CoA to glucose formation? Succinyl coenzyme. Now succinyl coenzyme is nothing but an intermediate of. So what could be the next reaction? Succinate. Succinate থেকে fumarate. Fumarate থেকে আগে ম্যালেট, তাই না? ম্যালেট থেকে বাইরে এসে, ম্যালেট থেকে বাইরে আসবে, বাইরে এসে অক্সিডেট, ফসফেট পাইরুভেট, তারপর উল্টো দিকে গিয়ে ফ্রাক্টোজ 1,6 বিসফসফেটেজ, পিএইচআই, গ্লুকোজ 6 ফসফেটেজ। তাহলে এই যে রিয়াকশন লিখলাম, এই রিয়াকশন দিয়ে আসতে পারে। এছাড়া প্রোপিনল কোত্থেকে আসে? এই তিনটে অ্যামিনো অ্যাসিড ব্রেকডাউন করেও প্রোপিনল করে আসতে পারে, তাই না? There is only one amino acid which is purely ketogenic. Which one? Leucine. Rest all are either both glucogenic and ketogenic or only glucogenic. So except leucine, all amino acids must produce glucose. Suppose there are other amino acids like proline, like histidine, like arginine, like glutamine, GLN. All these by several reactions will produce glutamate at some point in their catabolic pathway. তাদের ক্যাটাবলিক পাথওয়ে কোনো না কোনো পয়েন্টে গ্লুটামেট তৈরি করবে। Once the glutamate is formed, it is transaminated to form alpha ketoglutarate. Right? In this reaction, this is one amino acid, so one keto acid, say for example oxaloacetate or pyruvate will be converted to aspartate or alanine. If the enzyme is utilizing oxaloacetate, then the name of the enzyme will be if the enzyme utilizes pyruvate, then the name of the enzyme will be if you are not writing with me, you are losing something. If you have just realized you should have written, you have lost that. Follow? So these are the ways by which different amino acid can enter into the formation of this thing. Is there any other amino acid which enter through fumarate? Phenylalanine and tyrosine. Very good, very good. Sabas. Sona chile. ঠিক আছে? Now these pathways, why these pathways are so important? Why? কেন ইম্পর্টেন্ট কেন? এত ইম্পর্টেন্স কিসের এই পাথওয়ে? আরো অনেক কিছু আছে, তাই না? আরো অনেক কিছু আছে, এই পাথওয়ে দেখানো, রিঅ্যাকশনগুলো দেখানো আছে এখানে, দেখে নাও। এটা দেখো আর শোনো, why these pathways are so important. These pathways are so important because our body mainly rely on glucose for survival. All right. Now, when we get glucose, we need insulin for utilization of that glucose. In a clinical condition, when insulin is deficient, again, what is that? Diabetes mellitus. Where insulin is deficient, then whole metabolic system is jeopardized, because now stories have changed. Previously it was glucose who was feeding, now who is feeding? Ketone bodies, breakdown of fat. The blood is laden with glucose, but nothing can be used. Most of the cells cannot utilize it, or more precisely, adipose tissue and skeletal muscle cannot utilize the glucose without insulin. So they always feel hungry, and that sense of hunger makes a person eat more and more. So blood glucose level further rises because it cannot be utilized due to lack of insulin. Now, in such condition, which pathway will be performing and which pathway will be shut down and which pathways will be bypassed? We have to rely, our cell is full of glucose, but cell is hungry. So what the cell will do? Breakdown of glycogen, but glycogen is not there, it is already over. Now, gluconeogenesis. So, in lack of insulin, gluconeogenesis becomes what? The only way to get glucose within the cell. Within the cell. So we have a lot of glucose, but still we are producing more glucose. এইটা একটু দেখো তো, there is a term called futile cycle. Futile means not useful at all, no use. So can I call this gluconeogenesis and glycolysis, these pathways together, as futile cycle? Because breakdown of glucose produces 2 ATP, whereas production of glucose from the same pathway require 4 ATPs. So it is actually energy wasting process. Should our body continue this process? Yes. Yes, because it is producing glucose when there is no one who is giving glucose inside the cell. Inside the cell, there is no glucose. Cell is there, glucose is there outside the cell. Inside the cell, there is no glucose. It can only be produced from. But problem is, this pathway occurs only in liver, and liver does not require insulin for entry of glucose. Liver does not require insulin for entry of glucose. So then, who utilizes, who regulates the step? Who regulates the step? If only it can be produced by liver, it can be utilized by other tissues. The glucose by this pathway produced by liver, it can be utilized by other tissues, provided insulin is there. Otherwise, small amount may take place in kidney also, which may provide glucose. Correct? এই পড়তে হবে না। এই লাইনে লিখে রাখতে পারো। এটা আমি বলেছি গ্লাইকোলাইসিস পড়ানোর সময়, যা লেখনি যা লিখে রাখতে পারো, অলরেডি বিন ডিসকাসড। ওকে, একটা জিনিস মনে রাখবে, ইনসুলিন and glucagon. Insulin and glucagon are two such hormones who always and everywhere work as an antagonist. At least India and Pakistan fought together when? In Salman Khan's movie. You haven't seen? Tiger Abhi Zinda Hai? Isn't it? See? So at least India and Pakistan fought together for Salman Khan and Katrina Kaif. Okay? But insulin and glucagon, they are always antagonists. No other such hormones are there. Adrenaline, noradrenaline, no, not like it, not like this match. Never. Growth hormone, statins, no. Right? Only these two. You see, now this topic is very complicated actually. I don't know how much you are understanding. Many questions can be asked. Like the last question I asked, who decides that liver should produce glucose by gluconeogenesis? Who? If blood glucose level is low, then glucagon. If blood glucose level is high, then when blood glucose level is low, it is glucagon, correct? Huh? But when blood glucose level is high, like in case of diabetes mellitus, then no. The availability of free fatty acid, is a sign that body is starving for glucose. Availability of free fatty acid. In absence of insulin, free fatty acid content in the blood increases, which triggers gluconeogenesis in liver, correct? Not directly, but through acetyl coenzyme A carboxylase enzyme. I told you it is very complicated. More you enter, more it becomes complicated. You must see the question on what basis they have asked. Simply, most of the answers you will get from our class, if you are careful enough to listen to and to make a quick note down then you can get most of the answers, 99.999% answers you will get. 0.1% I don't believe because I have not made the question paper. Right? So you'll get. Now the important thing. Gluconeogenesis cannot be completed without Cori cycle. First line is important. Cori cycle operates, but from last class I have been telling when we are asleep, when we are asleep, when we are asleep, then what is Cori cycle about? Formation of glucose from lactate. But I am saying that when you are asleep, when sleeping, we are not exercising, then who will do this work? Alanine cycle. So when we are exercising, Cori cycle. So Cori cycle is the actual process through which our muscles should be trained. A good athlete, a trained athlete is trained to utilize this Cori cycle very well, and we are just opposite. See, very fit athlete. How many fit athlete you have seen like me? Who walks just little faster than a snail? Have you seen any athlete walking so fast, little faster than snail? No. So because I am not trained, but you are. During the sports day, you will become more trained only for one day, and next day half of the population will be in pain. For one day, they will be jumping, dodging, everything will be there, but next day half of the school, half of the population will be in the hostel. Ooh, that is a problem. So throughout the year, if you train, you can train your muscle to utilize this Cori cycle very well. Here skeletal muscle, liver, blood. Muscle produces glucose oxidized to pyruvate, pyruvate to lactate, anaerobic glycolysis. Lactate is transported back to liver via blood. In the liver, lactate is converted to pyruvate. Pyruvate enters mitochondria. Inside mitochondria, pyruvate is again converted to malate. Malate comes out of mitochondria. Outside mitochondria, malate is converted to oxaloacetate. From there, phosphoryl pyruvate. From there, glucose. That glucose is released, expelled from the liver. Why? By the grace of presence of one enzyme, glucose 6-phosphatase. Without this enzyme, liver will look like muscle, because muscle has no glucose 6-phosphatase enzyme. Liver has. That's why liver is liver, muscle is muscle. Right? Liver can release the glucose produced in it. Number one, because of presence of glucose 6-phosphatase. Number two, what is another reason? Due to glucokinase. Since glucokinase has very high value of Km, these two properties helps liver to release glucose from liver. These two properties helps liver to release glucose from it. Right? সেম জিনিস, দুটো করে ফসফেট বেশি লাগছে। এগুলি এখন আসে না, আগে আসতো। Cancer biology. During cancer, the cancer tissue utilizes a lot of energy for rapid division, cell division. So they require a lot of energy and they rely on Cori cycle to produce more energy in presence or absence of oxygen, doesn't matter. See, cancer cell glucose to pyruvate to lactate, lactate to whatever. Same pathway will occur in cancer. So cancer cell mimic what? Exercising muscle. But it is not muscle. It can be a glandular cell, it can be brain cell, it can be bone cell. Huh? But their metabolism will be like this, because this particular cycle is necessary to produce energy, supply of glucose. Okay. Alanine cycle, whereas one difference, let me draw, otherwise you will say, sir didn't study and that's why didn't tell me. Looks like liver or muscle? Liver, na? Muscle. It's liver, na? And this is muscle, na? Okay. Breakdown of protein produces alanine. Alanine by transamination produces pyruvate. Pyruvate by pyruvate lactate dehydrogenase forms lactate. Lactate is released and transported to liver via blood. Lactate is converted to pyruvate by lactate dehydrogenase, same enzyme. Pyruvate to glucose, pathway already told. Glucose is then released. It doesn't go to alanine, it goes to pyruvate, and again this cycle goes on. Wherever some portion is missing, the alanine will supply the amount of pyruvate. And alanine comes from the breakdown of protein. And this cycle occurs when we are sleeping.